Next, we have another speaker, Dr. Mark Christian Felipe R. Redillas. He is a associate professor at De La Salle University, also the biology department. He earned his PhD in biological sciences, an MS in environmental engineering and biotechnology in Mongi University, South Korea. Sorry. He received his BS biology major in microbiology at the University of Philippines in Los Baños. Dr. Redillas has been involved in the study and development of drought-tolerant transgenic rice, as well as those with improved nitrogen use efficiency. He is involved in the development of N-molecular sensors, nitrogen molecular sensors, to monitor the nitrogen status of rice. He is a member of the Philippine Society of Microbiology and a current board member of the Teachers Association of the Philippines, or BIOTA. Welcome, Sir Mark, and let's take it away.
Thank you, po. We'll just share the file right now. Good morning. My name is Mark Christian Felipe R. Redillas from De La Salle University, and I'll be talking about molecular-based nitrogen monitoring system in rice towards smart farming and agriculture. So nitrogen exists in different forms in the environment. Actually, 78% of the air that we breathe is made up of nitrogen gas. But it is the soluble form in ammonium and nitrates that are utilized by plants. These are the ones that are readily available for them for use. All of which can be a contributor in pollution if they're not properly regulated. These guys can cause respiratory and heart disease. And N2O, or the nitrous oxide, actually is 300 times more powerful than CO2. Not only it affects our health, but it actually depletes the stratospheric ozone. So nitrogen, if not properly regulated and controlled, is not good for us and for the environment. When we grow plants, there will be several factors that are considered and involved for reaching maximum yield. So the type of cultivars that you use, the climate should be appropriate for the cultivar, the machineries used for plowing, the water availability, of course the fertilizers and the machineries. All of these affects the yield. In addition to this, there are also major abiotic problems that significantly affect the harvest in the field. You have the drought, the flood, salinity, and even the fertility of the soil, which at least among these four, the fertility, that's the one that we can augment or improve. The deficiency or oversupply of these nutrients can actually be harmful to plants. The case of alkalinity and aluminum toxicity, calcium deficiency, copper deficiency, phosphorus and potassium, and more importantly, nitrogen. So either deficient or there's an oversupply, it's not good to the crops. In the case of nitrogen, which is the target of this study, plants that are grown in limited amount of nitrogen or no nitrogen, we'll see here that it is stunted. It has yellowish color and they're actually narrow, which again limits the grain production. If you see this, the plants in the field are actually visibly yellow if they don't receive enough or the optimum amount of nitrogen. So if it's too low, the panicle formation will be reduced because there will be less tillers, and of course low tillers will give you low panicles. Low panicles give you lower grains, thus giving you lower amount of harvest. And at the same time, if you put too much nitrogen, it's also not good since plants grow quickly. They're too weak, and it's also very prone to infestation. So that's why we need to look at smart farming and also called precision agriculture. And it's all about growing backed up by data and constant monitoring of your crops. For example, if there's too much or too little nitrogen can be alleviated by looking at these different steps that we can do to reduce excess nitrogen and also improve limited nitrogen use. So even if there's just small amount of nitrogen that we provide, as long as we are able to monitor the growth properly, then we can say that plants are receiving enough amount. Because not 100% of the fertilizer that we give is absorbed, actually less than 50% is absorbed by plants, and most of these will just go to runoff and thereby polluting the receiving bodies of water. In 2016, there was a report published in the Philippine Institute for Development Studies where it says the recommended application rate of fertilizer is 100 kilograms per hectare, but what was applied was 74 and 73 in irrigated and dry and wet seasons, respectively, and at the same time in rain-fed farms, it's way lower than that. And of course, what we do here is we expect what is going to be the yield, which is way lower than the maximum or expected yield. So reasons were high fertilizer price and lack of water. So the question is, what if we don't really have to give them 100% according to this suggestion, since most of the nitrogen are going to be wasted anyway? So what we just need to do is actually support or consider this smart farming, where we monitor our crops closely and responding to this data we get appropriately. So several methods on how nitrogen is monitored is either through destructive or non-destructive method. Destructive will be collecting tissue samples and analyzing it in the lab. Thereby, later on, you'll have an idea or how much nitrogen or that your plant contains. The non-destructive method utilize a different point of view on how to monitor plants. For example, in SPAD, it relies on the greenness. The stronger the color green will give you a higher value of SPAD, meaning plants are healthy, but it doesn't mean it is good for them. Just like in rice, it can lead to lodging with very weak comp. HandDP relies on chlorophyll fluorescence, which relies on the photosystem 2 activities related to the photosynthetic machineries. LCC, or the leaf color chart, also relies on the colors of the rice leaves and will actually give you a suggestion on how much fertilizer you need to provide to your plants. So in most developed countries, they're already using drones and the Sierra sensors, where it relies on leaf reflectance, which collects images from the field and process it in softwares and give you an idea or suggestions on how much fertilizers you need to put or augment. But how do we make this real time? Why? Because from this point, we think that the responses of these sensors are delayed, which means when plants are exhibiting yellow in color, we know that they're suffering already, which means plants are already exhibiting symptoms because they're already suffering from factors or stresses from the environment. So that's why we need to development of sensors that are required. So what we think is to rely on these transcripts in rice. So we started with plants exposing them to nitrogen starvation for several days, and per day we collect tissue samples from roots and leaves and have them analyzed for next generation sequencing, or the RNA-seq. And we'll see here differentially expressed genes. It's a lot. And from that, we filtered some of the genes that are responsive to or specific to nitrogen by doing a separate round of RT-PCR. And then if we got specific genes, we do cloning and do rice transformation, of course, testing it in the lab. So first product of the test was receiving this number of transcripts that are actually involved in nitrogen metabolism, responding to reduction of nitrogen, in this case until 7 days. Here is the shoot and roots and so on. These are all known genes that are involved in nitrogen metabolism. Now, through filtering and looking at the nitrogen metabolism pathway, several of these genes were able to respond to of nitrogen, but our attention was caught by these two genes, the ALN and UPS1. If you see here, there's a negative N, which means the nitrogen was removed for 10 days, and the expression of ALN is actually increasing, while if we put them back in nitrogen containing media, their expression was repressed. In opposite, the UPS1 is downregulated when there is no nitrogen, and its expression was expressed really, really high when they're placed in nitrogen-containing media. Now, these genes are actually involved in ureate metabolism, or in the purine metabolism pathway. Purine metabolism pathway is involved in the production of ureates. Ureates are the main pathway or nitrogen metabolic pathway in nitrogen-fixing plants. This must be very important because rice are not ureate formers. They're amide formers, meaning they synthesize amino acids. But for nitrogen-fixing plants, they convert the ammonium fixed here in the bacterioid, converting it to amino acids directly, or after converting it to amino acids, producing these ureates. While at the same time, in purine metabolism, it's converted right away to allantoin. Now, we also tested that allantoin can actually be used by rice as a sole nitrogen source. And we have a separate paper for this showing or indicating how allantoin overproduction in rice were able to improve the nitrogen use efficiency of rice. So here we isolated the promoter of these specific genes, ALN and UPS. Also isolated the coding sequence of the gene. So we isolated promoters and the CDS and inserted it in a gene cassette containing the reporter protein luciferase. So you have the promoter driving the expression of the CDS and at the same time the luciferase. The purpose of luciferase is used for testing the activity of the promoters. So since our genes are responsive to no nitrogen and with nitrogen in the medium, we can test this by attaching the promoter to this reporter gene, okay, reporter proteins. And it will actually give a higher fluorescence right after it comes in contact with the substrate luciferin. So after this, we produce the transgenic plants through tissue culture and using the agrobacterium transformation method. So here are examples of the transformants or transgenic plants containing these sensors. Let's just call this ALN plants and this one's UPS plants. Now here basically what we can see is if we grow them in medium containing high amount of nitrogen and with low amount of nitrogen, the UPS exhibited this fluorescence and non-fluorescence after spraying with luciferin as the substrate of the luciferase protein. On the other hand, ALN plants, as expected, didn't have expression of luciferase if the nitrogen is provided, but if there's no nitrogen in the medium, they express the luciferase. Though this is rapid and it's a real-time monitoring system, disadvantage would be it's very expensive because you have to produce transgenic plants and imaging system. This one is just another setup where it was incubated for a longer time, as well in 5 days and of starvation and negative, and the same result were obtained. Now, the current application of the transgenic plants that was produced in the lab is they underwent mutagenesis. And looking at these mutants that are either hypersensitive or insensitive to nitrogen starvation or over supply of nitrogen. So it depends on their expression, and that this will be filtered and will be analyzed for either for SNP analysis through NGS or our functional analysis and identification of phenotypes if they're exposed to genomic editing through Cas9 or CRISPR-Cas9. So, overcoming the problem of expensive production of sensors, we tried to improve this into a step wherein it can be done in the field and on-site by using the concept or principle of loop-mediated isothermal amplification. We have to note that these genes are only responsive to nitrogen signal. So when the plants detect through their roots that there is a minimal amount of nitrogen in the soil or even in the medium, you can actually monitor them through their shoots. So, and these are nitrogen specific. So since we already know the sequence of these genes, we are going to target the transcript of these genes and look at the expression or turbidity later on. Basically, it will look like this, depending of course on the dye that we use. A higher expression means higher turbidity and deeper or stronger dye colors, while lower expression will be look like this. Now, this is not from our study, but just to give you an example of how it could probably look after the assay. So, takeaways why we do smart farming is actually this is nutrient specific, which means the response that the farmers will do is right to what the problem is all about. Okay, it actually theoretically achieve maximum yield because you're not going to waste resources trying or testing it for other sources of problem. There will be lowered operation cost, increased product quality, can actually save time, and of course, very important is to reduce environmental footprint and of course, water conservation. So I'd like to acknowledge my professor and my PI for the success of this study, as well as the institutions where these were done, and for my current project here in De La Salle University. Thank you very much, and once again, good morning, and I'll see you later at the Q&A portion. Thank you. Doc Mency, naka-mute po.
Okay, don't go away first, Sir Mark. I just want to ask you one question before we go to the next speaker, and then we'll go to the Q&A. In your sa tansya mo, in your perspective, if we do come up with a on-field site-based sensor, how what do you call this? How easy will it be to be able to come up with that based on your previous experience in other places? And how do you think will it be passed on or cascaded to the farmer? And this is only for rice,
Yes
right?
po.
Rice, para sa rice lang. So, sa tingin mo, pag nagawa mo to or how you're doing it in Lasal, mga what are some of the hurdles or what are some of the things that we can look forward to?
Sa sensor development po, no? Since we are not really targeting or producing transgenic rice in this type of sensors that we're currently doing here in LaSalle, so I think it will be easier to implement this or apply this in the field. Ang problem kasi nung sa Korea back then is the sensor was made in transgenic plants. Dapat naka-embed yung sensor dun sa genome ng plants, they were producing transgenic plants. And similar din sa Philippines, yung acceptance din ng local community with GMO sa Korea, I think is the same. Meron laging resistance from the local communities. Although labs, maraming labs doon na really, in the top tier pagdating sa molecular biology research. But when it comes to application, no, or deployment ng research sa field talaga, medyo may resistance sa community. So, our lab, meron siyang field na from other universities too na may special agreement sila. And so from that local community, meron ding resistance na nangyayari from time to time. So yun po, I think it's very common. Actually, it's always discussed during meetings on how we can implement and how the farmers can, readily accept kung paano yung sa mga GMOs nga po. But here, that's why one of the objectives of this sensor development, especially coming back here sa Philippines, knowing na mas medyo ano yung mga Filipinos pagdating sa GMO, nanununog ng field. Yun, approach would be to minimize on transgenic production and develop yung technology that can be easily deployed in the field. Sa ginagawa namin with my graduate student ngayon, si Miss David, is yung approach is to yung design ng sensor is yung madali siyang gamitin sa field. And basically just to get a sample from the rice plants and then just put it on a small tube. And hopefully, ganun lang siya kabilis, theoretically speaking, and as yung expectation. But we still have to test that pagdating sa field. Pag sa lab kasi, probably it will work and it's going to be a different story when it comes to testing in the field.
Yeah, so really good thing there is to have the farmer scientist communication and connection early on. So you can do this. Thank you, Sir Mark.
Thank you very much.
So we're gonna come back with you in the Q&A later to be led by Dr. Amalin.